Our lesson for today is on the central dogma. So go ahead and title your page. And the central dogma is this idea that our cells contain DNA, and all cells contain DNA, all living things, and that DNA is going to code for our proteins. And then those proteins are going to cause our traits or our characteristics. So what this first arrow represents is it represents the DNA coding for the protein. And then the proteins, they cause our traits. And we have seen this already because we learned that DNA codes for HLA proteins, and those HLA proteins result in our ability to identify self versus non-self cells in our body. So our DNA coded for those proteins, and those proteins resulted in our traits. So let's take a look at a cell. Not all cells are round, but we tend to draw them as round because most of the time we're talking about animal cells, and a lot of times animal cells are pretty round. Now, inside of the cell, there's a nucleus. And then there are ribosomes. And all cells are going to have ribosomes. And all cells are going to have DNA. Now, in eukaryotic cells, that DNA is found inside the nucleus. But in something like a prokaryote, their DNA is going to be just inside of their cell because they don't have a nucleus. So we're talking now about this DNA. That's what the central dogma boils down to. It boils down to the DNA. So when we talk about DNA, we often talk about it in terms of chromosomes. And a chromosome is what we see under the microscope when it's all coiled up. And it often looks something like this. And then we also draw them replicated. So you might be used to seeing chromosomes where they look something more like this, with this centromere in the middle. So this is a chromosome. And chromosomes are made of DNA. And this is a chromosome. They're sister chromatids because it's a replicated chromosome, so it's going to share the same DNA. Now, if I zoom in on this chromosome, if I take this DNA, I can stretch it out and it has this shape, this particular shape that's called a double helix. And what I've drawn, these two lines, these are actually the backbone of the DNA. And the backbone are the individual DNA strands. And each individual DNA strand has bases in between the two strands. It's kind of hard to draw it on that one because it's so narrow. This one will be a lot easier. So I'm going to represent these bases with lines. And some bases are longer than others. Trying to draw them sort of equally spaced out here. And so inside of the double helix, it is held together by these bases. And so the backbone is on the outside, and then bases are on the inside. I'm going to draw the bases in terms of their letters. So we'll draw a couple here just to kind of get us going because there's not a lot of space. But then what we have is we have bases that are A's, and they're going to always pair up with bases that are T's. And then we've got bases that are C's, and they are always going to pair up with bases that are G's. And these can go in either order any way you want, but a G is always going to pair up with a C on the other side. And if I have an A on one side, I'm going to have a T on the other side. And these can go 2 T's in a row, 8 T's in a row. It doesn't really matter, as long as you always have what we call complementary base pairing. And then when we get back down here, we can just draw again our bases. These bases, they're going to be held together with hydrogen bonds. And so I'm going to make just some dots to represent the hydrogen bonds. And I'm drawing 3 dots between any C and G, because there are 3 hydrogen bonds between those, and then 2 dots between A's and T's to represent the 2 hydrogen bonds between those bases. Okay, so this whole structure is DNA. And that's what chromosomes are made of. And then a section of DNA, if I had, say, from right here to say right here, this section of DNA, that's what we call a gene. So let's define some things. Let's start by defining a gene. And the reason that I want to define a gene is because a gene is going to relate specifically to our protein. So genes are section of DNA that codes for a specific protein. And we should remember this from when we talked about our HLAs, because on chromosome 6, there was a gene for HLA-A, and that's going to code for a specific HLA-A protein that then lives on the surface of the cell. Not all proteins live on the surface of the cell, but HLAs do. Now, when I'm talking about this code, when I say that it codes for, what I'm really talking about is I'm talking about the order and the number of the bases. And another way to talk about order and number is to say the sequence. So in this gene, I would have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, let's call it 12 bases. And the order that they go in, the A, C, G, T, T, C, that is going to be the code that determines which protein is made. And that's what most of this unit is going to be about. It's about how we interpret that code in order to make the protein. But let's back up and just talk about some characteristics of DNA. And we've talked about these before, but we're going to put just the main ones in our notes today, just to make sure that we have them all in one place. So the first characteristic of DNA that we're going to make sure that we always know is that it has a double helix, which is the shape. And that is this twisted shape with 2 strands making up that double helix. So it's a double helix, that's the name of the shape, and it is 2 strands make up that double helix. That's what we mean when we say double helix. It's a helix shape times 2. So we're always talking about 2 strands. Now, those DNA strands, the individual strands, are made of nucleotides. What do I mean by a nucleotide? Well, nucleotides are made up of a sugar, specifically a sugar called a deoxyribose. And that sugar has this sort of a shape. And that deoxyribose sugar has a base attached on one side. It has a phosphate attached on the other side. And the bases that we're talking about here are, we're talking about the A, the T, the C, and the G. And as we saw in our model, the bases are paired or complementary. I always spell this wrong. And what we mean when we say that the bases are paired or complementary is that there's the A base, or adenine, is always going to be bonded to thymine, which is T, and cytosine, which is C, is always going to be bonded to guanine, which is G. I put the correct number of hydrogen bonds here, but we are going to go into that in much more detail later. But let's go ahead and add that. So the strands of DNA are held together with hydrogen bonds. So let's do a quick review. DNA has this double helix shape. It's this shape right here. And that shape is made up of a backbone, and then bases are in the middle. Each backbone plus base is called a nucleotide. So the backbone is the sugar and the phosphate, and then that base is the CT or AT or whatever, and that is going to, in combination with each other, the backbone and the base are going to make up a nucleotide. A section of DNA is called a gene, and the order of the bases in that gene is going to code for a specific protein. And then depending on what the protein is, that protein is going to result in our traits.